Hi, my name is Brandon Ubesha, and I am an undergraduate researcher in the Lorne Andrews Lab. And I will be presenting my project, developing a biophysical mathematical model to optimize repression using CRISPR interface for high throughput functional genomic studies. Biofilms are communities of microorganisms which attach to and colonize surfaces. Biofilms are harmful to humans since it's difficult to destroy them due to their extracellular matrix. And it also promotes infections in the body and on medical devices, such as catheter-associated infection. Current methods such as antibiotics to decrease biofilm development on biomaterials have been increasing antibiotic resistance and do not fully prevent biofilm formation. CRISPR interface could be used as a potential solution since it can be used to target biofilm infections through gene slicing and allows us to understand more about biofilm formation. CRISPRi regulates gene expression. It utilizes a dCas9 and sgRNA complex. It represses genes by blocking transcription initiation or elongation. sgRNA compete for common dCas9. The complex binds to target DNA through sufficient complementary and require polar spacer adjacent motif PAM sequence. There has been previous work done on CRISPRi systems by the Chen Research Group. Synthetic genetic circuits have shown their potential in a number of applications like energy medicine and more. Sharing dCas9 has been included in models of CRISPRi-based circuits. Their effects on circuits behavior haven't been fully explored though. This group made a model that accounts for sharing limited amount of dCas9 protein for arbitrary CRISPR-mediated gene expression and found as complexity of CRISPR-mediated gene regulation networks increase, limited dCas9 becomes a bottleneck. In this project, our objectives are to understand the effect of altering the copy number of the target gene as well as expression of dCas9 and gRNA genes on output repression, and then to refine the parameters to improve agreement with the experimental results. Research plan has 5 steps. First, do a sensitivity analysis on the model to predict how altering the copy number and expression of the CRISPRi components, then to tune the expression of the gRNA and dCas9 to optimize repression. 3 is to create the model with inducible promoters. 4, do a sensitivity analysis of gRNA concentration, and then to fit the model to experimental data obtained from the flow cytometry for a more accurate model. So what we've done so far is we made a sensitivity analysis of the dCas9 and sgRNA, as well as an inducible promoter. We were able to recreate source competition CRISPR-mediated gene repression model based off the model from the Chen group. And then we were able to modify repression model to include an inducible promoter via MATLAB. And as you can see here, at 1 and 0.5 ATC, lowering the concentration flattens out the graph due to a smaller decrease. And for graphs 0.1 and 0.5, you cannot see the S-curve for inducer concentrations in the designated ranges for the X and Y axis. And what was also done was a sensitivity analysis of gRNA concentration. And then progress so far. So we've done a surface plot for sgRNA production rate. And alpha D production rate of dCas9 and the total concentration of the ith DNA target. And we did a surface plot to see how repression changes as DIT is increased, and sgRNA production rate increases, and production rate of dCas9 decreases. And as you can see here, as DIT increases for each of the surface plots, while sgRNA production rate and production rate of dCas9 increase, there becomes more of a dip in repression. And for some of our current and future research, right now we're trying to get a more accurate model because it's a little bit off of our experimental data. So we are going to fit the experimental data obtained from the flow cytometer, and in the hopes of creating a more accurate model that we could use in the future. And that is my presentation. Thank you.